Hi, good evening. Hi, good evening, everybody. I'm back with you for today's second lecture. So, before we can go to the next step, let me give a recap with the keywords of photosynthesis today, right? And once that is through, we'll go into the three major cycles of photosynthesis. The first one would be glycolysis, right? And details on that. Before that, let me give a recap through keywords. And there will be photosynthesis with keywords. I'll give you the terms and then I'll give you the explanation of the words in short, right? So here's what we go. The first word would be photosynthesis, which is a process where plants make carbohydrate using sunlight, energy with carbon dioxide, water, and chlorophyll, right? And the second keyword would be light stage of photosynthesis. The definition has to do with the production of hydrogen ions to form NADPH, which I'm sure you all are aware of what the extension of the word is. It's an abbreviation of NADPH, right? So NADPH would be nicotinamide adenine dinucleotide phosphate, right? And the third keyword would be the dark stage of photosynthesis, right? So that would be the reduction of carbon dioxide, right? By the addition of hydrogen ions to glucose. That's the third keyword. The next would be cyclic phosphorylation. And that has to do with the pathway 1 in photosynthesis, which produces ATP, right? From the energy released from high energy electrons, which are then returned to the chlorophyll. The next keyword would be photolysis. Please note down these words or make a recording of everything that I speak, because this is absolutely academic stuff. It's good for those preparing for the NEET exams and those preparing for the National Eligibility Test for the UGC CSIR, right? Exams. In the NET exams, it would be life sciences, whereas in the NEET exams, this would be biology, right? So this is part of the botany part of it, which I'm speaking right now. Okay, going back to the keywords, the next word would be chloroplast, which means a cell organelle that contains chlorophyll, right? And the next would be chloroplast, which I repeat again, is cell organelle that contains chlorophyll. And the next would be chlorophyll, which is the pigment that absorbs light. The next word would be autotrophs, organisms that make their own food. Leaf, site in plant where photosynthesis takes place. Thylakoid membrane, site in the chloroplast where pigments are located and the light reactions take place. The next keyword you need to remember is electron transport system. It occurs in the cristae of the mitochondria. Carrier molecules accept high energy electrons from NADPH and transfer them to other molecules. In the process, energy is released, which is used to make ATP. What's ATP? Adenosine triphosphate, okay? That's an energy molecule. The next word would be light-independent reactions. Second stage of photosynthesis is not dependent on light, but is controlled by enzymes. Stroma is the next keyword, fluid in chloroplast where carbon dioxide is produced, right? The next keyword would be factors which affect photosynthesis are carbon dioxide, temperature, and light, right? Last but not the least, let me just spell out the formula for photosynthesis. In short, it will be carbon dioxide plus water plus light in the presence of chlorophyll, which releases or leads to the formation of glucose and oxygen. In chemical formula, it would be 6 molecules of carbon dioxide plus 6 molecules of H2O plus light in the presence of chlorophyll that gives you glucose, which is chemically known as C6H12O6 plus 6 molecules of, or 6 atoms of oxygen, right? So that's it about the keywords of photosynthesis, and next would be the key cycle of glycolysis, which is one among the first cycles in photosynthesis. So what's glycolysis about? All about glycolysis or glycolysis, and the details onto it. So glycolysis basically is a set of reactions that converts glucose to pyruvate or lactate. This is the first metabolic pathway to be elucidated, and hence is considered as, any idea? Considered as what? I'm going to just elaborate that part to you, right? As a paradigm of metabolic pathways, right? Start off. So glycolysis is also called what? Any idea? Let me just mention this word to you. Emden-Meyerhoff pathway, right? So the keywords in glycolysis are going to start with this. Emden, E-M-B-D-E-N hyphen M-E-Y-E-R-H-O-F-F pathway. The complete set of reactions occurs in the cytoplasm of virtually every animal cell. The entire process occurs without molecular oxygen, which means it's an anaerobic activity, right? Right, now, okay. We'll go into the details later. Now, about the details of whether it's an anaerobic or aerobic and all these things, I'll explain it to you as we go up to the chapters. Okay? Glycolysis consumes 2 ATP and generates 4 ATP. Okay? Thus, the process results in the generation of 2 net ATP. The process also generates 2 NADH. What happens to pyruvate depends upon the presence or absence of mitochondria in the cell, or upon the availability of oxygen in mitochondria containing cells, which means glucose, 2 ADP consumed, 4 ADP generated, leading to what? The generation of 2 NADH plus hydrogen ions, whereby it leads to the formation of 2 pyruvate plus 2 atoms of hydrogen. So in erythrocytes, RBC, cornea, lens, there are no mitochondria. So where pyruvate goes to lactate. Cells with mitochondria are relatively lack of O2, anaerobic conditions, right? And enough O2, you have aerobic conditions. So when I said lack of availability of oxygen mitochondria means it's anaerobic, isn't it? So the more the relative availability of oxygen leads to an anaerobic condition. In such cases where it's anaerobic, you have pyruvate going to lactate. Where it's enough of oxygen, it's an aerobic condition, you have pyruvate leading to the formation of carbon dioxide and water, right? For the glycolytic pathway to continue, NAD plus has to be regenerated. In erythrocytes, no mitochondria, and in mitochondria possessing cells under anaerobic conditions, NAD plus is regenerated from NADH during the conversion of pyruvate to lactate, right? And the formula would be CH3COCOO in the presence of NADH plus a hydrogen given an NAD plus, which leads to the formation of lactate, which is CH2CHOHCOO negative, right? Right. In mitochondria, possessing cells under aerobic conditions, NAD plus is regenerated by either malate aspartate shuttle or alpha glycerylphosphate shuttle, which transfer the reducing equivalents from NADH into mitochondria for electron transport chain, thus regenerating NAD plus in the cytoplasm, right? Now, these are the keywords that you need to always remember, followed by alpha glycerylphosphate shuttle and aspartate shuttle, right? We have the malate aspartate shuttle, alpha glycerophosphate shuttle. Then we have the electron transport chain, the glyceraldehyde 3-phosphate, glucose, and the G6-phosphate, fructose, right, 6-phosphate, and F1,6-biphosphate, dihydroxyacetone phosphate, and then goes to the glyceraldehyde 3-phosphate, 1,3-biphosphonate, biphosphate, glycerate, and phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and then pyruvate. Now, these are the keywords. Okay. After these keywords, you need to remember the enzymes, which act as a catalyst to move the action. So let's spell out the actions, the reaction first. Glycolysis consists of two phases. In the first phase, you have glucose, which is broken down into 2 molecules of glyceraldehyde, 3-phosphate, in a series of 5 reactions. In the second phase, another series of 5 reactions. Convert these 2 molecules of glyceraldehyde 3-phosphate into 2 molecules of pyruvate, right? Phase 1 consumes 2 ADP, and phase 2 generates 4 ADP. The net ADP production in the entire process is 2, right? So now, we need to know the procedure, right? See, once I finish a description of this process, I'll show you a diagram, okay? And when I show you the diagram and start explaining, you will not see me speaking, but you can hear me speaking, because otherwise your attention would be focused on me rather than looking at the board, right? So when I explain the diagrams, I'll focus the camera onto the diagrams, the illustrations, explain it to you. And once both of these is through, we'll wind up that subject. And then what happens is, towards the end of each week of lecture, we put in MCQs, okay? Keywords, illustrations for you to figure out how far you've understood this subject, right? You need to know these subjects in detail. So this is about biology. I think I read out the portions yesterday. For those who've seen my video yesterday, just go back to the video there. It's already posted there on YouTube. You can watch it. If you've still not understood what it's all about, connect with me on my WhatsApp number, 91 88 66 12 51, or my email address, which is newagedentalrehab@gmail.com, right? Get back on that and I'll explain things to you, if just in case you've not understood, right? All right, now for the diagrammatic representation, let me spell it out first for you, and then when I finish the lecture, I'll get back and show you the diagrams where it stands, right? So we have D-glucose, dextroglucose, get in the using a molecule of ATP, where it's converted to ADP, it's converted to glucose 6-phosphate. Now remember, this step uses an enzyme. What's that? Note it down. Hexokinase, okay, and glucokinase in the liver and pancreatic B cells, right? Now this gets converted to glucose 4-phosphate, which gets converted fructose 6-phosphate, okay, utilizing another molecule of ATP, which gets converted to ADP. So these are all investment reactions, right? Remember that. Here the reaction is catalyzed by an enzyme called phosphoglucose isomerase, right? So you have the product called fructose 6-phosphate, which gets converted fructose 1,6-biphosphate, right? Right? So that is number 3. We're going to number 3. Number 3 is in the presence of 6-phosphofructo-1 kinase, PFK1, otherwise known as PFK1, right? This produces fructose 1,6-biphosphate, which now goes in for the aldolase reaction, which is called the aldolase enzyme, which converts this fructose 1,6-biphosphate to dihydroxyacetone phosphate, right? And glyceraldehyde 3-phosphate, right? Now, in the presence of what? In the presence of triose phosphate isomerase. All right, that gets converted to 1,3-biphosphoglycerate in the presence of, that is glyceraldehyde 3-phosphate gets converted to 1,3-biphosphoglycerate in the presence of what? Note it down. Glyceraldehyde 3-phosphate dehydrogenase. These are all the ACEs, they are all enzymes that catalyze the action, right? And next would be the formation of 2-phosphoglycerate from 1,3-biphosphoglycerate in the presence of phosphoglycerate kinase, right? Remember these points, right? And then in the presence of phosphoglycerate mutase, right, 3-phosphoglycerate would be converted to 2-phosphoglycerate, right? Now, in the presence of enolase, this gets converted to phosphopyruvate. Okay, it liberates a molecule of water and phosphoenol pyruvate. And the next step, utilizing another ATP, we have the conversion of phosphoenol pyruvate to pyruvate in the presence of pyruvate kinase. So you have 10 products in the presence of 10 enzymes that are catalyzing the action, right? So remember these things, right? Now, the next is, I'm going to go into the individual reactions. Reaction number 1. Note it carefully. Note it. Hexokinase and glucokinase are enzymes that trigger the reaction number 1. Right? So, glucose, dextroglucose, in the presence of hexokinase and glucokinase, okay, which are a liver enzyme and beta pancreatic cell enzyme. These two make use of one ATP to convert that to ADP by one energy molecule being consumed, and you have a product called glucose 6-phosphate. So, in this reaction, this is the first intracellular reaction of glycolysis, and remember, all reactions are in the cytoplasm, so which means it's anaerobic, right? Requires an ATP, Mg. This is one of the investment reactions. Why? A molecule is used for the production of, glucose, right? Right. So the phosphorylation of glucose traps the glucose inside the cell, right? The reaction is considered as irreversible. What's the meaning of irreversible? It cannot be reversed. It doesn't come back, right? Good. And the next point is hexokinase has a constant for glucose of less than 0.1 mm, high affinity. It is also inhibited by the product glucose 6-phosphate, right? The liver hepatocytes and pancreatic beta cells contain another enzyme, glucokinase, which has a constant for glucose for about 10 mm low affinity. Glucokinase is not inhibited by its product glucose 6-phosphate. Glucose is induced by insulin. Glucokinase, correct me, glucokinase is induced by insulin. The levels of glucokinase in the liver of untreated type 1 diabetics are lower than normal. Note these points. NEET exams, MCQs, right? I'll just repeat that. Liver hepatocytes and pancreatic beta cells contain another enzyme called glucokinase. It's there. Glucokinase is found in liver hepatocytes and pancreatic beta cells. Could be an MCQ. Right. Glucokinase has a what? Km is the designation for glucose of about 10 mm low affinity. All right. So this is all about affinity. And glucokinase is not inhibited by its product glucose 6-phosphate. Glucokinase is induced by insulin. So the levels of glucokinase in the liver of untreated type 1 diabetics are lower than normal. So that's where people have the general addiction about carbohydrates, excessive carbohydrates, and the conversion isn't happening, and then you will become a diabetic. That's where they specifically say levels of glucokinase in the liver of untreated type 1 diabetics are lower. So the conversion is less. So the action of glucokinase is less. So the conversion goes less. So what happens? The formation of glucose 6-phosphate, or the breakdown of glucose, is less. That's when you become diabetic. Next, hexokinase and glucokinase are isoenzymes. Therefore, irrespective of the isoenzymes catalyzing the reactions, the KQ, the delta G and delta G0 for the reaction remains the same. I'm specifically saying these terms because you get to note them down and clear it up that these are known to you. Verify them. The reaction number 2 in glycolysis is phosphoglucose isomerase enzyme triggers that reaction, where from the second product, glucose 6-phosphate, the enzyme phosphoglucose isomerase converts to fructose 6-phosphate. This reaction is readily reversible, not a controlling step, and functions in both glycolysis and neogenesis. Conversion to an aldose to a ketose, right? So that's the second reaction. The third reaction is 6-phosphofructo-1 kinase, or the PFK1, or phosphofructokinase 1. All right? What happens here? The fructose 6-phosphate, in the presence of this enzyme, 6-phosphofructo-1 kinase, and the use of 1 ATP molecule, which gets converted ADP as energy, another investment reaction, we get the product fructose 1,6 biphosphate. Excuse me. And to explain the reaction, this reaction is the rate-limiting step of glycolysis. It is irreversible again, and the committed step where it is an allosteric enzyme and also a major regulatory enzyme. We have invested a second ATP molecule here. The fourth reaction is about aldolase reaction. So what's aldolase reaction? That's an enzyme that causes the fructose 1,6 biphosphate, which was formed before, to be converted to dihydroxyacetone phosphate, right? Plus glyceraldehyde 3-phosphate. Now, when I show you the diagrammatic representations, make a recording of these things because you need to know the formulas properly if you're going for the NEET exams and the NET especially for the UGC exams, right? Okay. Now, the next one, to explain the reaction, we have now 2 phosphorylated trioses. Only glyceraldehyde 3-phosphate is used in glycolysis. Note that point. Only glyceraldehyde 3-phosphate is used in glycolysis. Therefore, dihydroxyacetone phosphate has to be converted into glyceraldehyde 3-phosphate. This occurs in the next step. Remember, okay? All right. So the next thing is we have dihydroxyacetone phosphate being converted to glyceraldehyde 3-phosphate in the presence of the enzyme triose phosphate isomerase. Right? Here what happens? Triose phosphate isomerase catalyzes the interconversion of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So because of the interconversion, one glucose molecule can be converted to 2 glyceraldehyde 3-phosphate molecules, right? And the sixth reaction is glyceraldehyde 3-phosphate dehydrogenase. So what happens here? Glyceraldehyde 3-phosphate in the presence of glyceraldehyde 3-phosphate dehydrogenase enzyme, and PI and NAD gets converted to NADH and H+, that's hydrogen, to give 1,3-biphosphoglycerate, right? Here what happens? The enzyme oxidizes the number 1 carbonaldehyde and then adds a phosphate to the group. Okay, phosphate group. We have an acid anhydride in the product, 1,3-biphosphoglycerate. Remember from bioenergetics that acid anhydrides are high energy bonds. The phosphate on the number 2 carbon is not a high energy bond. We have used an NAD plus for the oxidation reaction. The cell has limited amounts of NAD plus. So somewhere along the line we have regenerated or glycolysis will stop. This reaction is a target for arsenate, ASO4 3 negative, right? The arsenate resembles inorganic phosphate, PI. PI stands for inorganic phosphate. In the presence of arsenate, the product of the reaction is 1-arseno-3-phosphoglycerate. I repeat, 1-arseno-3-phosphoglycerate. This production is unstable and decomposes into arsenate and 3-phosphoglycerate with no ADP formation. After this step, glycolysis continues. The enzyme contains an essential thiol, Thiol, thiol. In brackets, you could write it as cysteine-SH group at the active site. Iodoacetic acid, ICH2COOH, is an inhibitor of this reaction. I repeat, iodoacetate acid, ICH2COOH, is an inhibitor of this reaction. It reacts with the active site SH group and inhibits the enzyme. And we have reaction 7S next, which is phosphoglycerate kinase controlled. So 1,3-biphosphoglycerate in the presence of phosphoglycerate kinase and using an ADP, which gets converted to ATP. Okay, kind of a reduction reaction, I guess. And we have 3-phosphoglycerate. It's an addition reaction. Okay, and you have, this is the first step of energy production where ADP becomes to ATP. This is referred to as a substrate level phosphorylation action as opposed to oxidative phosphorylation that occurs in the mitochondrial ATP production. We have recovered both ATP that were invested. Now, remember that each glucose gives 2 phosphoglycerate molecules. Another point for the MCQs. Right? And next would be the reaction number 8 with the phosphoglycerate mutase. What happens here? 3-phosphoglycerate in the presence of phosphoglycerate mutase gets converted to 2-phosphoglycerate. And reaction number 9 is enolase, where we have 2-phosphoglycerate in the presence of enolase, liberating a molecule of water, giving phosphoenol pyruvate. Here in the reaction, it catalyzes. Okay, this enzyme catalyzes the dehydration of 2-phosphoglycerate to form phosphoenol pyruvate, PEP. Right? Recall from the bioenergetics lecture that PEP contains a high energy bond. Okay, this energy is inhibited by fluoride. Right? And the 10th reaction would be pyruvate kinase controlled. So phosphoenol pyruvate in the presence of pyruvate kinase gets another addition with another ATP being formed to the formation of pyruvate. This catalyzes the transfer of the phosphate from PEP to ADP to generate ADP and pyruvate. Again, this is substrate level phosphorylation, right? This reaction completes the part of glycolysis. This reaction completes that part of glycolysis that is common to both anaerobic and aerobic metabolism, right? Under aerobic conditions, in presence of mitochondria, pyruvate can enter the citric acid cycle. This citric acid cycle will be the next on discussion tomorrow while I finish glycolysis, the aerobic and anaerobic. So far I've been speaking about aerobic. Now next we go into the anaerobic glycolysis, right? Let me just finish with the last portion of the aerobic glycolysis. Under aerobic conditions, in presence of mitochondria, pyruvate can enter the citric acid cycle. NAD+ is regenerated by malate and aspartate shuttle or by alpha glycerophosphate shuttle, right? Under anaerobic conditions or in the presence of mitochondria, pyruvate is reduced to lactate by the following reaction and regenerate NAD+. Anaerobic glycolysis. When pyruvate cannot be oxidized within mitochondria, for some reason, example hypoxia, genetic defects in pyruvate dehydrogenase, or citric acid cycle enzymes, genetic defects in electron transport chain, pyruvate is reduced to lactate by lactate dehydrogenase. Right? Pyruvate. Okay? In the presence of lactate dehydrogenase, H plus NADH and NAD plus is formed, right? We have lactate. Right? And the synthesis of 2,3-biphosphoglycerate. Alright? All cells contain a catalytic amount of 2,3-BPG, biphosphoglycerate. Red blood cells contain a high concentration of 2,3-biphosphoglycerate, relevant to 4 mm. It facilitates the release of oxygen from hemoglobin. So about 20% of glucose goes through this shunt in RBC. No net ATP is generated if 1,3-BPG is converted to 3-PG, phosphoglycerate, via 2,3-BPG, by phosphoglycerate, if the anti-glycolytic pathway in the RBC occurs. RBC is a red blood cell, occurs via the formation of 2,3-BPG. Glycolysis will yield no net ATP because the 2 ADPs produced by pyruvate kinase will be equal to 2 ADP consumed in phase 1 of glycolysis. Right? The next is 2,3-biphosphoglycerate levels in erythrocytes increase in high altitude where partial pressure of oxygen is low. These are all MCQ answers, right? Note them. I repeat that, 2,3-BPG levels in erythrocytes increase in high altitude, like mountaineering, things like that, where partial pressure of oxygen is low. Loss of function, mutations in pyruvate kinase increases the level of 2,3-biphosphoglycerate and RBC, thus shifting the sigmoidal curve of oxygen binding to hemoglobin to the right. I repeat, thus shifting the sigmoidal curve of oxygen binding to hemoglobin to the right. If loss of function mutation occurs in any of these enzymes upstream of the step involved in the formation of 1,3-BPG, the levels of 2,3-BPG and RPC, red blood corpuscles, will be lower than normal, thus shifting the curve to the left. I hope you note that. Now the control of glycolysis. It's obvious that glycolysis must be controlled, right? Enzymatic control can be exercised by 3 different common methods. One is the allosteric effectors, 2, covalent modification, and 3, transcription of enzymes. Allosteric effectors means the transient binding of molecules, to the enzyme. To change the conformation, effect is observed in milliseconds. And in covalent modification, general phosphorylation effect in seconds. And a transcription of enzyme effect is observed in hours. There are only 3 major points of control of glycolysis. And the points of control are hexokinase and glucokinase reaction number 1. I repeat, hexokinase and glucokinase reaction number 1. Hexokinase is found in most tissues. It has a low Km for glucose, less than 0.1 mm. Allows for the phosphorylation of glucose when blood levels of glucose are low. This permits tissues that rely on glycolysis extensively to carry out the phosphorylation. And hexokinase is strongly inhibited by its product, glucose 6-phosphate G6P. Liver parenchymal cells, hepatocytes, contain 1 isozyme of hexokinase, glucokinase, and a high Km for glucose at 10 mm. It talks about affinity, high affinity, low affinity, and those. Right? And the concentration in cell reflects blood concentration. Liver uses glucose at a significant rate only when blood glucose is elevated. Glucokinase is not inhibited by G6P. It is inhibited by fructose 6-phosphate F6P, which is generated by reaction number 2, stimulated by a fructose 1-phosphate, which is F1P. Regulation occurs via inhibitory protein. F6P stimulates the binding of the inhibitory protein. And F1P stimulates the dissociation of the inhibitory protein. And glucokinase is inducible in liver by insulin. CAMP, adenine monophosphate, decreases glucokinase levels in liver. Glucokinase is also present in pancreatic B cells, where it plays an essential role in glucose-induced insulin secretion. Because of the low affinity, this enzyme serves as a sensor of blood glucose to coordinate insulin secretion appropriately. Let me repeat that. Because of the low affinity, this enzyme serves as a sensor of blood glucose level to coordinate insulin secretion appropriately. The next is the phosphofructose 6-phosphofructose-1 kinase, of the PFK1 reaction number 3. This is the rate-limiting step of glycolysis. First committed step of glycolysis, subject to the greatest degree of regulation by allosteric effectors. Positive effectors, the pi and AMP are positive effectors of PFK1. The AMP is a negative factor of F16-BPAH, which catalyzes the reverse reaction converting F16BP into F6P and pi. Fructose 2,6 biphosphate. This is a positive effector of PFK1 and a negative effector of F1 and 6-BPH. Negative effectors. ATP is an allosteric inhibitor of PFK1. Next one, citrate. Glycolysis and citric acid cycle are coupled via PFK1 because citrate, an intermediate in citric acid cycle, is an allosteric inhibitor of PFK1. When the citric acid cycle is saturated with high levels of citrate, citrate leaves mitochondria via a transporter, a tricarboxylate transporter, and inhibits PFK1. This prevents generation of pyruvate, which feeds into the citric acid cycle. And the next you have is the Pasteur effect. Oxygen inhibits glycolysis, right? So the consumption of glucose in the presence of oxygen generates much more ADP. The inhibition is probably due to the inhibition of PFK1 by ADP. And the next is the pyruvate kinase reaction, which is number 10. It's inhibited by high concentrations of ADP. Isoenzyme found in liver is activated by fructose 1,6 biphosphate. Liver enzyme is subject to phosphorylation, active in the dephosphorylated state, inactive in the phosphorylated state. Inactivation by phosphorylation is a function of CAMP, dependent protein kinase in liver. Enzyme 4. The enzyme is inducible by high carbohydrate concentrations and also high insulin levels. And next, the control of 6-phosphofructose-1 kinase PFK1 by fructose 2,6 biphosphate, regulation by CAMP. Let's talk about its effect in liver. Fructose 2,6 biphosphate behaves like AMP. Positive effector of 6-phosphofructose-1 kinase PFK1. The positive effect involves an increase in the affinity of PFK1 for F6P and also a decrease in the inhibitory effect of ATP. Negative allosteric effect of fructose 1,6 biphosphatase. This compound is not a product of glycolysis. In fact, its only known function is as a regulator, is found in all cells, which F16 biphosphatase. Hormonal control of fructose 2,6 biphosphate in liver. Glucagon, the presence of glucagon receptor, and ADP being converted to CAMP, tri going to mono, decrease in fructose 2,6 biphosphate and inhibition of glycolysis. Right, and with this explanation, I'll wind up this portion. There's a little extension, more of an extension, which will be continued in the next class. Okay, continuing. Glucagon, a 29-AE hormonal peptide, is released by the alpha cells of the pancreas and interacts with the glucagon receptor on the surface of the liver plasma membrane. The binding of glucagon to the receptor is sensed by adenylate cyclase enzyme located on the inner surface of the plasma membrane. So adenylate cyclase catalyzes the conversion of cytosolic ATP, triphosphate, to monophosphate, CAMP and PPI. Right? CAMP ultimately causes a decrease in the levels of fructose 2,6 biphosphate. And CAMP acts as a second messenger. And note, CAMP acts to inhibit glycolysis in liver, but in most other cells, CAMP stimulates glycolysis. The difference is that liver is the primary organ which controls blood glucose levels. Liver can make glucose, gluconeogenesis, as well as degrade glucose is glycose as needed. CAMP stimulates gluconeogenesis in liver and inhibits glycolysis. CAMP induces decrease in fructose 2,6 biphosphate levels and is at least partly responsible for these effects. In some cells, CAMP actually stimulates the synthesis of fructose 2,6 biphosphate, example heart muscle. In skeletal muscle, CAMP does not affect the synthesis of fructose 2,6 biphosphate. And we just go into the synthesis of fructose 2,6 biphosphate. Okay, in fructose 2,6 biphosphate, Synthesis is from fructose 6-phosphate, an intermediate in glycolysis, via the enzyme 6-phosphofructo-2-kinase PFK2. This is not the same as 6-phosphofructo-1-kinase PFK1. Fructose 2,6-biphosphate is destroyed in cells by the enzyme fructose 2,6-biphosphatase. Both of the above enzyme activities are located on the same protein. It is a bifunctional enzyme and is referred to as 6-phosphofructo-2-kinase fructose 2,6-biphosphatase. CAMP is responsible for the regulation of fructose 2,6-biphosphate levels in liver. So the PFK2 F2 6-BPase is regulated by protein kinase A, a phosphoprotein phosphatase, as follows. And the diagrams will be shown to you once I finish the class today and tomorrow. Each section, there's glycolysis or citric acid cycle or whatever. When I finish it, then I will get back to the diagrams and then I'll put in questions for you to answer, right? So phosphorylation causes an activation of the active site responsible for the synthesis of fructose 2,6-biphosphate, an activation of the site responsible for the hydrolysis. Dephosphorylation has the opposite effect. Increased levels of glucagon causes an increase in CAMP levels. The second messenger activates CAMP-dependent protein kinase. This phosphorylates 6-phosphofructo-2-kinase fructose 2,6-biphosphatase. The synthesis of fructose 2,6-biphosphonate is inhibited and its degradation is promoted. So a decrease in fructose 2,6-biphosphate makes 6-phosphofructose-1-kinase less effective and fructose 1,6-biphosphatase more effective. Glycolysis is inhibited. Insulin opposes the action of glucagon. Glucagon and insulin clearly act in opposition to one another, and the insulin-glucagon ratio of blood determines the intracellular levels of fructose 2,6-biphosphate and therefore the rate of glycolysis. Different tissues express different isoforms of the bifunctional enzyme PFK2 and PFK2K and F2,6-BPase encoded by different genes. The liver isoform possesses sites for protein kinase, a dependent phosphorylation, CAMP inactivates PFK2 and activates F2,6-BPase and thus reduces F2,6-BP levels. The heart isoform has phosphorylation sites for protein kinase A, but at different part of the molecules. CAMP activates PFK2 and has no effect on F2,6-BPase and hence increases F2,6-BP levels. The skeletal muscle isoform does not possess phosphorylation sites for protein kinase A and hence has no effect on F2,6-BP levels. Okay, and now here are some clinical considerations. A. Lactic acidosis. Write it down. Lactic acidosis, characterized by high blood levels of lactate, generally greater than 5 mm, while normal levels are usually less than 1.2 mm. So the blood pH can be less than 7.1 in severe cases. Most tissues can convert lactate to CO2 and H2O through the TCA cycle, tricarboxylic citric acid cycle, right? If the oxygen supply is inadequate, cells must rely on glycolysis for ATP production. A decrease in ATP enhances glycolysis at the level of PFK1 and produces more pyruvate and hence lactate. Accumulation of plasma lactate may be secondary to tissue hypoxia, circulatory insufficiency due to shock, heart failure, severe anemia, mitochondrial enzyme defects, and inhibitors of oxygen transport like carbon monoxide, cyanide, liver disease, and ethanol. Bicarbonate is usually administered to control acidosis associated with lactate overproduction. Dichloroacetate is used as a drug to treat lactic acidosis because it activates pyruvated dehydrogenase in mitochondria, right? And which facilitates the conversion of pyruvate to acetyl CoA so that lactate production is decreased. Strenuous exercise leads to anaerobic glycolysis. This causes overproduction of lactate. For those of you doing exercise, remember this. Don't over-exercise. Strenuous exercise leads to anaerobic glycolysis. breakdown of glucose, anaerobic. This causes overproduction of lactate. You can have problems. Diabetes is the next clinical situation. In diabetes, insulin activity is low. Type 1, no insulin. Type 2, insulin resistance. Consequently, glucagon levels increase. Thus, glucagon-insulin ratio is higher in diabetes. There is no change in glucose uptake in liver because this tissue does not express GLUT4. The insulin-responsive facilitative glucose transporter. So what's GLUT4? Is the insulin-responsive facilitative glucose transporter. In skeletal muscles and adipocytes, glucose uptake decreases because of the absence of insulin-dependent recruitment of GLUT4 into the plasma membrane. In liver, the glucose is not metabolized effectively because glucagon insulin is affected by glucokinase, CAMP, PFK1, CAMP, which brings down the effect on F2,6-BP, and PFK1, which increases the conversion F1,6-BPA. So this also results in an increase in gluconeogenesis, producing more glucose. Neogenesis refers to creation. And glucose from neogenic precursors, alanine and glycerol. These precursors come from muscle and adipocytes as a result of increased protein and amino acid breakdown and lipolysis to provide energy because of the decreased utility of glucose as the energy source. Next is glucokinase deficiency. Mutations in glucokinase gene leading to inactivation of the enzyme are associated with a form of non-insulin dependent diabetes mellitus type 2 called maturity onset diabetes of the young, MODY. Complete absence of the enzyme activity will lead to type 1 diabetes because of the lack of insulin secretion in response to blood glucose. This condition is associated with severe hypoglycemia, right? Mutations in the gene leading to increased activity of the enzyme will cause hyperinsulinemic hypoglycemia. I repeat, hyperinsulinemic hypoglycemia. And the next is pyruvate kinase deficiency and hemolytic anemia. Genetic defect causes a 5 to 20% reduction in red cell pyruvate kinase levels. It's rare, but the most common genetic disease associated with the glycolytic pathway results in markedly lower ADP concentrations in erythrocytes. Cells swell and lyse. Reticulocytes are unaffected because these immature red cells contain mitochondria unable to generate ADP through oxidative phosphorylation. The levels of 2,3-biphosphoglycerate are high in erythrocytes. No Heinz bodies, HEINZ bodies. The second most common genetic cause of hemolytic anemia, only next to glucose 6-phosphate dehydrogenase deficiency. What's that? G6PD deficiency. Make a note of those drugs that you can use for those with G6PD deficiency, okay? We'll mention that on a later stage when I come to the pharma section. All right? E is other glycolytic enzyme defects. Deficiencies in the activities of phosphofructokinase, phosphoglycerate kinase, phosphoglycerate mutase, and lactate dehydrogenase represent important genetic defects in glycolysis. All of these have common clinical features: exercise intolerance, myoglobinuria, hemolytic anemia, absence of lactate, increase in forearm exercise test, and increased glycogen deposition in the liver and skeletal muscle. Right? So that's it about glycolysis. I think I've given a good exposition on the whole thing. Just note down the points, right? And keep in touch while I get back to you tomorrow with the next cycle. That's the tricarboxylic cycle. That's a citric acid cycle, right? And now I'll just switch over to the screenshots. You won't see me, but you'll hear me speaking, and I'll show you the diagrams of what I spoke to so far, right? And that should give you an idea of a quick catch-up of what I've been trying to speak to, and try to recap what I've told you. And note the points, keep the recording safe, don't delete them. They'll be useful for your NEET exams and for the UGC CSIR NET exams. So all details coming in for your effective answering of all questions. Remember each point by point, and you should be a winner. And with regard to photosynthesis, read up the keywords that I mentioned today and relate them to the present procedure, and you should be okay, right? I'll switch over now.